Chapter 21, the digestive system. This is actually a pretty long chapter, and I usually would spend 2 days in lecture just on this. The first day just reviewing the anatomy, because there's so much to remember from the digestive system. So we're going to start by reviewing some of the anatomy. So why do we have a digestive system? It boils down to nutrients. Our body needs building blocks to help heal tissues, to help make proteins, to make enzymes, all the things that it needs to do. It needs nutrients. Well, how do we get the nutrients? Some can be produced by the liver. However, most you have to get from the external environment, so you have to be able to find a way to break it down to its smallest individual parts so that you can absorb it into the body, and then there has to be a mechanism in place to eliminate what is not used, and that's what the digestive system does. And it starts in the mouth with the oral cavity. We have the mouth and the pharynx. Remember, the main part of the digestive system is the alimentary canal. And the alimentary canal is basically one long tube. It starts at the mouth, ends at the anus. So it starts with the oral cavity in the mouth and the pharynx. Inside the oral cavity, we'll go over some pictures here in a minute, but inside the oral cavity we have the teeth to help grind and tear things up into little bits. And then we have saliva and salivary glands, which are accessory glands for the digestive system. The saliva is either mucose saliva or it is a watery saliva, but it has some enzymes in it to break down some carbohydrates. So carbohydrate digestion begins in the mouth. And then the mucose part of the saliva is a binding saliva and helps keep the food stuff together. So even though you're chewing it to break it down, you want to swallow it as one mass, and that's what happens in the head in the cephalic phase. But we'll get to that later. But then you swallow it. Whenever you chew up food and mix it with saliva, it's called a bolus, B-O-L-U-S. And then if we continue down the GI tract, after the oral cavity, the mouth and pharynx, the food passes into the esophagus. The esophagus is the tube that connects the pharynx to the next part, which is the stomach. So then we have the stomach, and which stomach leads to the small intestine. Small intestine leads to the large intestine, and then after the large intestine, it's eliminated through the anus, whatever was not digested and absorbed. Anyway, then there are valves and sphincters. These valves connect certain parts. For example, the valve between the esophagus and the stomach, that would be the cardiac valve. And then the valve between the stomach and the small intestine, that would be the pyloric valve. And the valve between the small intestine and the large intestine, that would be the ileocecal valve. And then there are two valves associated with the anus and their anal canal. There's the involuntary internal valve, and then there's the voluntary external valve. We'll get into that eventually, the sphincters, as we go through the alimentary canal. So there are accessory organs as we go through the alimentary canal that will secrete digestive enzymes that help with the digestion. I already talked about the salivary glands a little bit. We'll have a picture here in just a minute, but in the salivary glands there are three. We have the parotid or parotid salivary gland, which is the largest of the salivary glands, and then there's the submandibular salivary gland, and then sublingual salivary gland. The parotid salivary gland produces a mostly serous, S-E-R-O-U-S, saliva, which means it's full of enzymes, and these enzymes break down carbohydrates. The enzyme is called amylase. So it's salivary amylase to break down amylose, which is a carbohydrate. So carbohydrate digestion begins in the mouth. The submandibular salivary gland, it's a mixed salivary gland, so it's part serous and part mucose. So it does have some digestive enzymes in it, plus it also has the binding mucus saliva. And then the sublingual, which are going to be underneath the tongue, those salivary glands are mostly mucose salivary glands, so mostly produces a binding saliva. And then we have the pancreas. The pancreas is an endocrine and an exocrine organ. We talked about the endocrine function of the pancreas way back in the Unit 1. It has the alpha cells and the beta cells that produce glucagon and insulin to regulate your blood sugar. The exocrine function of the pancreas is to produce digestive enzymes. Most of the cells of the pancreas are called acinar cells, A-C-I-N-A-R. These acinar cells produce the pancreatic amylase, the pancreatic lipase, the pancreatic nuclease, the pancreatic proteases. These are enzymes that break down carbohydrates, proteins, and lipids. And then we have the liver. The only digestive secretion of the liver is going to be bile. We have bilinogen that comes from breaking down red blood cells, and then the bilinogen goes to the liver, part of it goes to the liver, and it can be converted to bile, and bile is used to break down emulsify fats. On a previous slide, I mentioned bolus. It's B-O-L-U-S, and bolus is going to be chewed up food mixed with saliva. You swallow the bolus. The bolus travels through the pharynx, it travels through the esophagus, and then enters into the stomach. Once the bolus enters in the stomach and it is mixed with the gastric juice, now it's called chyme. So C-H-Y-M-E is what we're going to call a bolus mixed with gastric secretions. It's kind of a soupy mixture. The chyme is going to travel through the stomach. Chyme enters into the small intestine, and then the chyme enters into the large intestine. And then the large intestine is going to absorb some water and electrolytes, and whatever is left over is no longer called chyme, it's called feces, and that's what is eliminated. And then we get to the large intestine when we get to the intestinal phase, because there are 3 phases of digestion: cephalic phase, gastric phase, and then the intestinal phase. In the intestinal phase, in the large intestine, there is some beneficial bacteria flora that help with a little bit more digestion in the large intestine. And that's what they mean by home to commensal microorganisms. These are bacterial organisms that help with digestion. That's not a part of you, it's just a part of your gut. So digestion begins in the oral cavity, and there's 2 types of digestion. There is mechanical digestion. Whoops. I really hate drawing with my finger. And then there is chemical digestion. All right, so mechanical digestion is physical digestion, breaking something apart, tearing it apart. And that's what your teeth do. Your teeth will grasp and then rip into whatever you're trying to chew up and to make smaller pieces. So mechanical is physical. Chemical digestion means using enzymes to break food down at the chemical molecular bond level. That's how we go from polysaccharides to monosaccharides, how we go from proteins to individual amino acids, how we go from lipids to individual fatty acids, using chemical enzymes to break the bonds. When we were going through the different parts of the alimentary canal earlier, we didn't mention the different parts of the stomach or the different parts of the small intestine. As we go through, we'll talk about those parts. Like when we get to the gastric phase, we'll talk about the parts of the stomach. And then we have a picture here. Let's go through the picture. So this picture on the left is showing all of the alimentary canal. So you put the food stuff into your mouth. That's called ingestion. And then you start chewing it. That is mechanical digestion. And then you have the salivary glands that are producing saliva, and then they create chemical digestion. All that happens in the oral cavity. Then you swallow. And as you swallow the bolus, it travels through the esophagus. The esophagus travels down through the thoracic cavity, passes through the diaphragm, and then enters into the stomach. So here's the stomach. Here are the different parts of the stomach here. There's a valve right here that's between the esophagus and the stomach. This valve normally stays closed. It's called the cardiac valve. It's also known as the lower esophageal valve, or sometimes it's called the sphincter. So it could be the lower esophageal sphincter or the cardiac sphincter, either way. It stays closed until the bolus gets real close to it and starts to touch it, and then it opens up, and as soon as it enters the stomach, it closes back again. It helps keep the gastric contents from going up into the esophagus. The gastric contents have a pH of about 2, maybe even 1, which is highly, highly acidic, and that acidic content could fry the tissues of the esophagus, and it causes an esophageal hernia or hiatal hernia. And some of you may have experienced that before. It's not fun. It causes all kinds of heartburn, and if it gets bad enough, it could start bleeding. Anyway, and then we have the parts of the stomach. We have the fundus of the stomach, which is for temporary storage of food. The first region the food hits is called the cardiac region of the stomach. Then we have the body of the stomach, which is the main part. And then we have the pyloric region. And then we have the small intestine. So there's a valve at the end of the stomach, this valve that separates the pyloric region from the small intestine. This is called the pyloric valve or pyloric sphincter. All right, so if we go back over here to the left, now we're in the stomach. And then the food stuff passes into the duodenum, which is the very first part of the small intestine. And then it goes through the jejunum, which is the second part of the small intestine. And then it goes through the ileum, which is the last part of the small intestine. And then it goes into the large intestine. Large intestine starts with the cecum, and then it goes to the ascending colon. Then it goes around the hepatic flexure. Then it goes over through the transverse colon. Then it goes around the splenic flexure. And then it goes into the descending colon. And then it goes into the sigmoid colon, and then it straightens out into the rectum. And then from the rectum it goes to the anal canal, anus, and then out. All right, the salivary glands, let's go over those again real quick that we did earlier. So here's the parotid, or the parotid salivary gland. It's the largest of the salivary glands, and it's in front of the ear. It produces an enzyme that's mostly serous, which means it's full of amylase enzymes to break down carbohydrates. This picture down here, this is the label for the submandibular salivary gland, which produces a mixed saliva, part serous, part mucose saliva. And then here's the sublingual salivary gland underneath the tongue, and it produces a mostly mucose saliva. Most digestion and most absorption occurs in the small intestine, so there are some adaptations to help facilitate digestion and absorption in the small intestine. One of those is called the plica circularis, which are going to be circular folds. If you look inside of the small intestine, it's not a flat tube. There are hills and valleys. Those circular folds are called plica. So instead of having a straight line, then it curves up and down this way. So that helps increase the surface area inside of the small intestine so that we can have as much room as possible, or as much surface area as possible, for more absorption. And then another modification to the lining of the small intestine is called a villi, or villus. It kind of looks like a finger-like projection that's sticking straight up. And then on top of the villus, there's also microvilli, which are hair-like extensions on top of the finger-like projections. And the microvilli have enzymes on them so that whenever they touch food stuff, as the chyme as it passes through, it will break it down. That's called contact digestion, touching it to break it down. In this picture on the right-hand side, it shows a section of the gastric mucosa, which is inside of the stomach. There are folds that we can see around here, but these folds are called rugae. So inside of the stomach, it's not a flat surface either. It has got folds in it called rugae. And then as the chyme fills inside of the stomach and the stomach stretches, then the rugae will flatten out. But it's another way of increasing the surface area inside of the stomach. Plus, in between each fold on the rugae, the gastric cells go down into what they call a gastric pit. So the gastric pit is full of cells that produce the gastric juice, like hydrochloric acid, like pepsinogen, like intrinsic factor. When we get to the intestines and we look at the intestinal wall, we have the plica circularis, which are the circular folds. We also have the villus and the macrovilli, but they also have cells that dip down towards what we call the submucosa, but they're not called pits, they're called crypts. So there's intestinal crypts and then gastric pits. This slide lays out some of the stuff that I was just talking about earlier. Modifications to increase the surface area. The stomach has rugae. The small intestine has plicae. They're just folds of tissue. And then in the small intestine, there are villi, which are finger-like projections. We don't find villi in the stomach, and we don't find villi in the large intestine, just in the small intestine. And then we have those cells that produce secretions that are emptied into the small intestine, into the lumen. In the stomach, they're called gastric glands or gastric pits. In the intestines, they're called intestinal crypts. And then we also have some submucosal glands that produce mucus. And mucus, not only is it a binder when it comes to saliva, but mucus is also a lubricant when we are lining the lumens of the alimentary canal. Plus, they're also a buffer. There's all kinds of bicarbonate in mucus, and that helps prevent the gastric juice or the enzymes from digesting your own tissues. The alimentary canal has 4 tissue regions or 4 tissue layers as it goes through the canal. The innermost layer, the layer that lines the lumen, the layer that is closest to the chyme or closest to the feces or closest to the bolus, is called the mucosa. The layer right beneath that is called the submucosa, and then on the next slide it's going to talk about the other layers, which is the muscularis layer, muscularis externa, and then the serosa. The serosa is on the outside. But let's start with what's on the inside, which is the mucosa. The mucosa has 3 layers to it. There's a mucosal epithelium, a lamina propria, and then there's a muscle called the muscularis mucosae. The mucosal epithelium, most of the time, is simple columnar epithelium. Most of the time is also ciliated, but the most common epithelium is going to be simple columnar, but there are some exceptions. In anywhere where there's going to be a lot of mechanical digestion, like in the mouth, the oral cavity is lined with stratified squamous. The oral pharynx is lined with stratified squamous. The laryngopharynx is lined with stratified squamous. And then the foodstuff passes, or the bolus passes into the esophagus. The superior portion of the esophagus is lined with stratified squamous, but then as we get down towards the cardiac valve and the cardiac sphincter, it slowly transitions into a simple columnar epithelium. Then it stays simple columnar through the stomach. It stays simple columnar through the small intestine. It stays simple columnar through most of the large intestine. When we get to the rectum, it transitions back into a stratified squamous epithelium, and then the anal canal is stratified squamous epithelium as well. The openings to the outside are lined with stratified squamous. On the inside of the mouth, stratified squamous, it's non-keratinized because it has to stay moist. Same thing with the tissues of the anal canal. Stratified squamous, it's non-keratinized because it has to stay moist. All right, the second layer of the mucosa is a connective tissue called lamina propria. There's plenty of lymphatic tissue in the lamina propria, especially in the gut, which are going to be your intestines. They're known as Peyer's patches. It's also known as GALT, G-A-L-T, which stands for gut-associated lymphoid tissue. In the anatomy book, they call it something different. Do you remember what that was? It was called MALT, M-A-L-T, which is mucosal-associated lymphoid tissue, so they are the same thing, gut-associated or mucosal-associated. Either way, it's the same type of lymphoid tissue. And then there is some muscle called the muscularis mucosae. This muscle helps to keep the alimentary canal kinked or coiled, especially in the small intestines. You have like 16 feet of intestines in the alimentary canal, and that most of that's going to be stuffed into the gut. So it's not a straight pipe, so to speak. It is coiled, highly coiled, and so the muscularis mucosae keeps it highly coiled. Plus, the muscularis mucosae will also help and assist with pushing foodstuff through the alimentary canal. All right, then we have the submucosa, which is the second layer of the GI tract, or a second layer of the alimentary canal. And the thing about that is mostly it is connective tissue, and this is where we're going to find a lot of the blood vessels and lymphatic vessels that to transport nutrients that have been absorbed. Plus, there's also part of the nervous system located here. We have a submucosal plexus. The submucosal plexus of nerves is for the muscularis mucosae muscle of the mucosa. So the last 2 layers of the alimentary canal wall, we have the muscularis externa, which we're going to start with in just a second, and then we have the serosa. So let's start with the muscularis externa. So there are 2 layers of the muscularis externa. There's an inner layer, and then there's an outer layer. All right, the inner layer is also known as the circular layer, because the muscle of the inner layer of the muscularis externa goes around the circumference. The circular layer of the muscle has to do with a certain type of movement. We're going to go over that later with a different picture. It's called segmentation. The circular muscles also create the valves and the sphincters, like the cardiac valve, the pyloric valve, so on and so forth. So the inner layer of the muscularis externa is the circular layer, it forms the valves in the sphincters. The outer layer is also known as the longitudinal, which means it goes along the length of the alimentary canal. Whenever the outer layer of the muscularis externa contracts, it propels the foodstuff forward. The propulsion of the foodstuff forward is called peristalsis. So the longitudinal muscle is responsible for peristalsis. The circular layer is responsible for segmentation. And there are pictures to demonstrate the difference between segmentation and peristalsis later. And then the outer layer is called the serosa. The serosa, for most of it, has another name. It is called the visceral peritoneum. If you think that sounds familiar, we've talked about these tissues in anatomy. The peritoneal layers. There's the visceral peritoneum, and then there's the parietal peritoneum. The parietal peritoneum being an outer layer, visceral being actually on the outer surface of the organ. If at any point on the outside of the alimentary canal it's not exposed to a cavity, then it is fixed in place. It's called an adventitia and not a serosa. This picture is showing you the wall of the small intestine. Now we can see the villi, the finger-like projections that stick straight up. And the cells that dip down in between each villi, they're in crypts, the intestinal crypts. The cells of the intestinal crypt will produce mucus, plus they also produce water, and they also produce some enzymes for digestion. So the four layers of the alimentary canal, we have the mucosa, which is closest to the foodstuff. Then we have the submucosa, and then we have the muscularis externa, where we have the inner circular muscle, outer longitudinal muscle, and then we have the serosa on the very outside, also known as the visceral peritoneum. Are the four basic processes of digestion together as a whole, and there's a picture later that helps also explain this if you're more of a visual learner. First, we have digestion, which we've already talked about. Digestion is breaking something down. And there's two basic types of digestion. There's mechanical digestion, and then there's chemical digestion. Mechanical digestion is physically tearing something apart. Chemical digestion is using enzymes to break it at a molecular level, breaking molecular bonds. Absorption has to mainly do with the direction that the molecules are going. Once the food has been broken down to individual molecules like monosaccharides, amino acids, and then fatty acids and triglycerides, they have to be taken into the body. Remember that the lumen of the alimentary canal is external environment. Until it crosses through a membrane to get inside the body, it stays in the external environment. So absorption, definition of absorption is moving molecules from the external environment of the lumen into the internal environment of your lacteals or capillaries. So that would be absorption, moving foodstuff from the external environment to the internal environment. And then we have secretion. With secretion, the movement is a little different. With secretion, the cells that line the lumen of the alimentary canal, if it secretes it into the lumen, so it's going from internal environment to external environment, that would be secretion. Plus, there are some paracrine secretions. That means the cells of the lining of the alimentary canal produce a secretion that interacts with the cells that are only surrounding it. And then also a secretion, it is moving ions from the cell of the lining and into your capillaries, into your cardiovascular system. And then we have motility. The types of motility are segmentation, and there's also peristalsis. Segmentation is a back and forth movement. It is used mainly for mixing the foodstuff with as much digestive enzymes as possible. So it does some mechanical digestion. And then peristalsis is a forward type of movement to propel foodstuff towards the end of the alimentary canal. Now, there are three challenges with digestion. Avoiding autodigestion. What does that mean? That means avoiding digesting your own tissues, because you have some protein enzymes. They're going to break down the proteins. Well, most of your tissues are made of proteins, and you don't want to break down your own tissues. When that happens, things like hernias, like hiatal hernias, heartburn, it happens. Ulcers happen. Mass balance, not leaving too much inside the body, so being able to eliminate excess, that would be the challenge. And then defense, because no matter how much you clean food, it's not as clean as you would like it to be, and definitely not as clean as the body would like it to be. So there has to be ways to fight against pathogens that is in the stuff that you put in your mouth. So for you visual learners out there, digestion, one of the processes is taking a big particle and breaking it down to individual particles, mechanical or chemical, either way. So digestion, breaking down to smaller parts. Digestion occurs in the external environment. It occurs in the lumen of the digestive tract. Now moving this small particle through the lining and into your cardiovascular system, this is absorption. So moving it from the external environment into the internal environment. Secretion is moving something from the cell into the lumen, that's secretion, or from the cell into the interstitial fluid that affects the receptors or binds to receptors that are closest to the cell, or it's going to secrete it through the interstitial fluid and into the capillaries. So that would be secretion. And then motility, moving it back and forth or moving it straight forward. This next part's a pretty easy read. We take in 9 liters of fluid a day. That's a lot. 2 liters are just from the food and drink that we consume. 7 liters comes from the secretions made by the stomach and the gut. Now, some of the secretions are what we call zymogens. Zymogens are inactive protein enzymes. The reason why the protein enzymes are released inactive, it is so that we don't auto-digest, to break down our own tissues. They're activated at a specific time whenever it's needed to break down the proteins that you've ingested and swallowed. So zymogens are proteins that need to be activated. So we'll learn how they're activated in the stomach and also how they're activated in the small intestine. And then we have mucus, which is a secretion, and mucus comes from goblet cells or serous cells or mucose cells. This mucus is a buffer, so it also protects the lining from the digestive juices. All right, some of this was mentioned earlier. Purpose of the muscles of the alimentary canal wall, the external muscles, the circular and longitudinal muscles, and the musculus mucosae. Well, moving the food stuff through the tract, that's just common sense. And then also to mechanically mix it, to break it into uniformly small particles. That's the segmentation that we were talking about earlier. Now, when it comes to the types of contractions, this is going to be familiar. We have tonic contractions and there are phasic contractions. So tonic contractions, just like tonic signals, they are constant. So the tonic contractions can last for minutes or hours. Usually, the sphincters are controlled tonically so that they can stay closed until we allow the valve to open to allow a little bit of food stuff to pass through into the next phase of digestion. And then we have phasic contractions, which just last for seconds, and that is more of a mixing type of motion for the muscles. Now there are cells called the interstitial cells of Cajal, named for Dr. Cajal. He's the one that found them. He is a Spanish doctor. Your intestines can actually move on their own, just like the heart can beat on its own without any interference from the nerve system. The intestines can generate their own peristalsis waves, their own movements. They don't need the nervous system to tell them to contract. And that has to do with their own system called the enteric nervous system, E-N-T-E-R-I-C. So the enteric nervous system can operate on its own. If you were to take the intestines out of the body, they would still move around a little bit to eventually, when they run out of oxygen and run out of glucose in order to create ATP. But the potentials that travel through the walls of the intestines are called slow wave potentials. The 3 types of patterns of contraction, there are 3 types of movements. There's peristalsis, which moves things forward. And then we have segmental contractions. Those are mixing movements. To mix food stuff up. So peristalsis and then segmental contractions. But then there's the migrating motor complex. Let me show what that looks like on the next slide. In the parts of the alimentary canal where there is no food stuff, like in the stomach if it's empty, or in the small intestines in those sections that are empty of chyme, they do something called the motor migrate or migrating motor complex. And it's a series of shutters or vibrations, and they help clean the inner lining, any kind of debris that might be left over. So there's a shuttering motion that goes through the stomach to propel stuff into the small intestine. And it only happens in those areas of the alimentary canal that are empty. If there is food stuff in certain parts of the alimentary canal, then those are the parts that do the segmentation and the peristalsis. Now let's look at the membrane potentials over here just real quick. We're not getting into any specific numbers. We see here that we have a little bit of depolarization occurring, and then it goes back down. So this is a sub-threshold. So this, if there is a threshold. So if the depolarization does not reach threshold, then there's no contraction. But then if it does reach threshold, then we have an action potential which travels along the alimentary canal for contraction, peristalsis, segmentation, stuff like that. Same thing. That can be added, so it's more like a graded potential. And then force of contraction. The more action potentials that occur in a small amount of time, the stronger the contraction. This picture is showing you the difference between segmentation and peristalsis. With peristalsis, the muscles in front of the bolus will relax. The muscles behind the bolus are contracted and propels the foodstuff forward. Remember, this inner lining is going to be lined with mucosal cells that help produce a mucousy secretion that's going to be kind of a lubricant to help also propel the foodstuff forward. With segmentation, it alternates contracting different parts of the circular muscles, because it's the peristalsis is responsible, or is from the longitudinal muscles. The circular muscles that will alternate contracting will push the foodstuff back and forth to mix it with as much enzymes as possible, plus it also does some mechanical digestion. It's like taking a tube of toothpaste and putting your hand in either side of the tube with the cap on, and then just squeezing your hands, alternating back and forth. The toothpaste inside the tube is going to go back and forth, and you can mix it. So like I was saying about the enteric system, it can act independently. It doesn't need the brain to tell it whenever it needs to contract and propel foodstuff forward. So the enteric system controls motility, it controls secretion, and then also controls growth of the digestive system. Some of the cells secrete their own neurotransmitters and their own neuromodulators. Those are the cells that are secreting secretions that only affect the nearby cells, the paracrine secretions. They also find that they have their own glial cells. Now, they don't know for sure exactly what they are, or they haven't named them yet, just that they do have their own supporting cells. So the enteric system is like its own nervous system. And there are two types of reflexes when it comes to the enteric system. There are short reflexes and there are long reflexes. The short reflexes start and end with the enteric nervous system. The long reflexes start with the central nervous system that affects the enteric nervous system. So this slide is showing you the long and the short reflexes. Short reflexes start in the enteric nervous system, and they only stay in the enteric nervous system if it starts with the brain or inside the spinal cord, which is outside of the enteric nervous system. Then that would be a long reflex. If we need to go anything more specific about this picture, we will later. Some of the more common hormones found in the GI system, the gastrointestinal system, like gastrin and cholecystokinin and secretin. Some of those enzymes we're going to talk, or hormones we'll talk about in depth when we get to these later sections of the alimentary canal. But the next slide breaks it down to a really nice table for you. Let's look at some of this. We have gastrin, which is a hormone that comes from the gastric pits of the stomach. The function of gastrin is to stimulate gastric acid secretion. It stimulates the stomach and stimulates gastric activity. So what causes the release of gastrin? Well, the presence of peptides or amino acids in the gut. We have receptors, and when the amino acids bind to the receptors, it stimulates the gastrin or the G cells to produce gastrin, which will stimulate gastric activity. Then we have cholecystokinin. Cholecystokinin comes from the cells of the intestinal crypts. And there are three different functions of the hormone cholecystokinin. It targets the gallbladder, and makes the gallbladder contract. It targets the pancreas, and it stimulates the release of pancreatic enzymes. And then it also goes to the stomach, and it inhibits gastric activity to slow it down so that we don't overwhelm the small intestine. The gastric contents have a pH of about 1 to 2, which is highly acidic. The pH of the small intestine is about 8, so it's way more alkaline. So there has to be time in order to buffer the acidic contents as soon as they empty into the duodenum, which is the first part of the small intestine. So until it can be buffered properly, we're going to slow down the stomach so that it doesn't send too much into the small intestine at once. All right, then we have secretin. Secretin comes from the intestinal crypts as well. It stimulates the pancreas to secrete bicarbonate. So it's going to secrete the buffer. So it'll buffer the contents coming into the stomach to about 8. The pancreatic enzymes work at a pH at about 8. And then secretin also inhibits gastric emptying, slows the stomach down. Then we have an enzyme or hormone called motilin. Motilin comes from the intestine, and that is the hormone that stimulates the migrating motor complex. That's the movement through the empty parts of the intestine to help propel things that are left over, detritus, little foodstuff, molecules that may still be left over that didn't follow along with the rest of the chyme. And then we have some I don't talk too much about, which is gastric inhibitory peptide and the glucagon-like peptide. And you can read what those do down here. The three main phases of digestion is going to be the cephalic phase. Cephalic phase. The cephalic phase is everything that happens inside of the oral cavity. The mechanical chewing, the chemical digestion from the salivary glands, the swallowing. All of that happens in the cephalic phase. We'll talk about in the next slide. And then the food stuff, when it passes into the stomach, we have the gastric phase. So we have all these secretions that produce gastric juice, which is part of the gastric phase. And then as the food stuff moves into the small intestine, or the large intestine even, that's all part of the intestinal phase. And we'll go over those parts when we get to the intestinal phase. So the cephalic phase is everything that happens in the head, the first phase of digestion. It starts with just smelling or seeing or even thinking about food. They call this a feed-forward reflex. It's preparing the stomach for the bolus that's going to pass through the esophagus. So if you ever smelled food and your stomach growls, or you see food and your stomach growls, it is preparing itself for the food that's about to receive. So this reflex starts in the brain, and the brain then sends signals to the stomach to help to start producing the gastric juice. And like we mentioned before, chemical and mechanical digestion begins in the mouth. Mechanical is chewing, using your teeth. But then we have the saliva for the chemical digestion. The enzyme for chemical digestion is called amylase, and it breaks down amylose, which is a carbohydrate. So carbohydrate digestion begins in the mouth. Plus, the saliva helps keep the oral cavity moist, and we need that for the chemoreceptors for taste. And then some of the saliva also has immunoglobulins in it to help break down pathogens that are still in the food that you are ingesting. The term mastication is the fancified word for chewing. So mastication is chewing, not to be mixed up with masturbation, which is a totally different process altogether. All right, I've mentioned this before, the three pairs of salivary glands. The parotid are the largest of the salivary glands, and it produces mostly a watery saliva, and it's full of enzymes. The more watery the saliva, the more enzymes there are in it for chemical digestion. And the submandibular salivary glands, they are mixed. It's part watery and part mucose. So it has some enzymes in it, but it also has some mucus-binding saliva. And then the sublingual salivary glands produce mostly a mucus-rich saliva, which is a binding saliva, so that you chew the food part, but then you also want to swallow it as one lump called the bolus. So the saliva is under autonomic control, so the autonomic nervous system. Which do you think, parasympathetic or the sympathetic nervous system? Well, salivary secretion, since it's resting and digesting, then that would be the parasympathetic nervous system, which is why when you go into the sympathetic nervous system, you get dry mouth. And that's what happens if you have to speak in front of a large group, or if you have to lecture about something that you barely know about. That creates no saliva in the mouth, and it gets real dry and makes it harder to talk. You may have experienced that when you're talking to somebody who is obviously lying through their teeth. They start to mumble, they start to say words wrong. It's because they're nervous and they're lying about something. Anyway, that's a psychology thing. Worry about that in psychology. All right, so then to swallow, deglutition is the fancy word for swallowing. So mastication is chewing, deglutition is swallowing. And when you swallow, it starts as a voluntary movement, but then the muscles that are skeletal muscles, mind you, they take over, and it's actually involuntary. So usually when we say skeletal muscles are voluntary muscles, there are some exceptions, and one of the biggest exceptions would be with the swallowing muscles, because once you start swallowing, it's automatic, but it's still using skeletal muscles. And then you have the epiglottis. The epiglottis is going to close over the glottis, close off the trachea, so the food goes into the esophagus and not into the windpipe. So this slide is showing you the steps of deglutition, or swallowing. When you get ready to swallow, the tongue pushes the bolus towards the pharynx. It stimulates the soft palate and the uvula, and it lifts up. And it closes off the nasopharynx, so that whenever you swallow, the bolus goes down and does not go up past the soft palate and out through the nose. Now, this typically does not happen. However, if you use air, force air up through the trachea when you're trying to swallow, it could dislodge the soft palate up, and then whatever you were swallowing would then come out through your nose. If you've ever laughed with liquid in your mouth, then you know what I'm talking about, because it comes out your nostrils. Okay, so the bolus then starts passing, and then it picks up on these different sensors that are back here, so that the next part happens. The larynx moves forward, the epiglottis moves down. The larynx moves forward in order to open up the esophagus, and then the larynx pushes down so that the bolus goes into the esophagus and not into the trachea. And then once it's in the esophagus, gravity takes over, and the bolus will then drop down towards the stomach and then be passed through the cardiac valve and into the cardiac region of the stomach. Now, the muscles of the esophagus can also do peristalsis. So if the bolus just doesn't happen to go down with gravity, and then we have the backup of peristalsis, the longitudinal muscles will then push the bolus down, which is how astronauts that are up in space that don't have the benefit of gravity, it's how they're still able to eat. The peristalsis will take over and push the food down. You can also stand on your head and drink a beer, like none of you have ever done that before. But anyway, if you do that, then the beer still gets to your stomach, right? All right, next slide. So the next phase is the gastric phase. This is all the stuff that happens in the stomach, and it's a pretty easy read. All right, there are three main functions of the stomach. One, we have storage. It stores the food stuff until it's ready to be processed by the stomach. The upper part of the stomach, which is called the fundus, or the fundic region. The fundus is where we have temporary storage of the food stuff, and that happens in the upper part of the stomach. And then we have digestion that occurs in the stomach, usually towards the lower part. Now we have some lipid digestion, but there is protein digestion. Protein digestion definitely begins in the stomach. And we'll talk about the enzymes that are used for that later. There is some lipid digestion that occurs in the stomach, but mostly lipid digestion occurs in the small intestine. So there is a gastric lipase that is released by the gastric cells or the gastric pits that will help break down some lipids, but most lipid digestion is going to occur in the small intestine. And then the third function of the stomach is defense. So these enzymes will break down pathogens that have made it past the oral cavity, gone down through the esophagus. Hydrochloric acid is highly acidic, has a pH of about 1 to 2, depending on how concentrated it is, and that's why the gastric contents have a very acidic pH. So digestion occurs with the cephalic phase. Now, this is the long reflex. Seeing food makes the stomach, or the brain stimulates the stomach to start producing the gastric juice to prepare it for the food that you're about to swallow and eat. All right, so you see the food, you smell the food, or maybe even you taste it when you put it in your mouth. Then the brain signals the stomach, the enteric plexus of the stomach, in order to produce gastric juice, which then will be secreted into the lumen to be mixed and prepared for the foodstuff that you are swallowing. Here's where we start getting into the good stuff in the stomach. The gastric secretions, those cells that produce the parts of gastric juice. And we'll start with gastrin. Gastrin is secreted by a cell called a G cell, and the G cells are found in the gastric pits. But gastrin stimulates gastric activity. So the stomach produces its own hormone to stimulate itself to produce more parts of gastric juice. And then we have parietal cells. Parietal cells are also found in the gastric pits, and they produce two types of secretions. They secrete hydrochloric acid, and they secrete intrinsic factor. We went over this in anatomy, so this is just a little bit of a review. Intrinsic factor is the biggest part of gastric juice, and it's very high, and I think it's one of the most important parts of gastric juice. Intrinsic factor allows you to absorb vitamin B12. Without intrinsic factor, you can't absorb vitamin B12. And vitamin B12 is vital. It's essential for making red blood cells and for making hemoglobin for red blood cells. So if you're deficient in vitamin B12, then you're going to be deficient in viable red blood cells, and it makes you anemic. You only get vitamin B12 from your diet. You can't get it from anywhere else in the body. It's not made by the liver. So you have to get it in your diet. For those people who have had gastric bypass surgery, back in the old days, they used to bypass the entire stomach, which means they bypassed the one part that you needed to produce intrinsic factor to absorb vitamin B12. So they would have to get vitamin B12 shots in order to not become anemic. Now, the hydrochloric acid is important to break down pathogens, but then the hydrochloric acid is also the activator for the zymogen. Earlier in the PowerPoint, we talked about zymogens. Zymogens are inactive protein enzymes. The protein enzymes are released inactive so that you don't digest your own lining. The inactive protein enzyme of the stomach is called pepsinogen, and that's what we're going to talk about next. So there are chief cells. Chief cells are found in the gastric pits, and they also produce two secretions. They secrete pepsinogen, which is the inactive protein enzyme, and they also release gastric lipase. So lipase is an enzyme that breaks down lipids, and this is where lipid digestion occurs in the stomach, but it's not that effective. It does work, but it's not that effective. The pepsinogen is activated pepsin because of hydrochloric acid. So hydrochloric acid cleaves the pepsinogen and turns it into pepsin, and the pepsin is the activated form of the protein enzyme to break down proteins. All right, so we also have enterochromaffin cells, or enterochromaffin-like cells, also known as ECL cells, and they secrete histamine, and histamine, like gastrin, stimulates gastric activity. So histamine comes from the ECL cell found in the gastric pits, and it stimulates gastric activity. We already went over intrinsic factor, and then we have cells called D cells. D cells are also found in the pancreas, and D cells secrete something called somatostatin, and somatostatin inhibits gastric activity. So the enzymes histamine and gastrin, or those hormones, histamine and gastrin, I should say, they stimulate gastric activity. Somatostatin inhibits the gastric activity to slow things down a little bit so that the stomach doesn't go too fast. And then the other cells of the other cells of the gastric pits are the mucose cells, and mucose cells produce mucus and bicarbonate so that your stomach doesn't digest its own lining. That can create ulcers. Let's take at this right-hand side, letter C, acid secretions from the stomach. So this is the parietal cell that's producing hydrochloric acid. This is how it does it. It takes water, and water is broken down to hydrogen and hydroxide. The hydrogen is going to go to the apical surface, and it's going to be exchanged for potassium. So hydrogen is going to go out towards the lumen, and then potassium is going to go in towards the inner part of the cell, and then potassium will go back out through the leak channel. So potassium can be used over and over, in order to pump the hydrogen into the lumen of the stomach. And then the CFTR channel right here. CFTR is for chloride. So chloride will then go down its concentration gradient into the lumen. So the hydrogen and the chloride come together to form the hydrochloric acid. Let's see what else is happening. The hydroxide, along with carbon dioxide from aerobic respiration, using an enzyme called carbonic anhydrase, will change it into hydrogen or bicarbonate. This is the formula for bicarbonate, and then bicarbonate is going to be traded for chloride. And then bicarbonate is going to go into the blood capillary. So whenever you eat a meal, the parietal cell, while it's making hydrochloric acid, is also pumping bicarbonate into your blood system, which is going to make you kind of alkaline. They call that an alkaline tide. There are 3 different triggers for the gastric pits to produce the gastric secretions. We have food entering into the stomach, or just seeing food, or just smelling food. This starts with the cephalic phase, or this is the long reflex from the central nervous system affecting the stomach. And then we have the presence of amino acids and peptides in the lumen. And then we also have the autonomic nervous system that is influencing the gastric secretions. All right, so all of those factors will then stimulate the enteric sensory nervous system, and it stimulates the G cell to produce gastrin. And then gastrin is going to stimulate the ECL cell, which produces histamine, which then stimulates the parietal cell to make hydrochloric acid. All right, to catch all that? So the amino acids stimulate the enteric nervous system, which then will follow this red line. The enteric nervous system stimulates the G cell. The G cell then produces gastrin. Gastrin then stimulates the ECL cell. Gastrin also directly stimulates the parietal cell. ECL cell produces histamine, and we mentioned earlier that histamine is a hormone enzyme that then also increases gastric activity by stimulating the parietal cell to produce hydrochloric acid. All right, let's see what else is happening here. If we follow the input of the vagus nerve also stimulates the ECL cell to produce histamine, and it also stimulates the parietal cell to produce hydrochloric acid. All right, now the hydrogen that is going to become hydrochloric acid is also binds to a receptor that stimulates the enteric nervous system. The presence of hydrogen in the lumen stimulates the chief cell. The chief cell releases pepsinogen, and the pepsinogen is changed by hydrochloric acid into pepsin, which is the active form of the protein enzyme. Also, the presence of hydrogen in the lumen has a negative feedback on the D cell. So it stimulates the D cell, which produces somatostatin, and somatostatin inhibits the G cell, so it stops the production of gastrin. Somatostatin also inhibits the ECL cell, so it stops producing histamine. And then somatostatin also comes all the way down this way and inhibits the chief cell to stop producing pepsinogen. All right, this slide's a pretty easy read. Now we're going to start with the intestinal phase, and this includes what's happening in the large intestine. All right, so the motility through the small intestine, we have the segmental and the peristaltic contractions. We went over that ad nauseum enough, so I'm not going to go over that again. There's also the migrating motor contractions. But the big thing about motility through the small intestine is that we want to make sure that it's not overwhelmed by the chyme going through. It takes about 6 to 8 hours for the chyme to move through the small intestine. Now, it passes through the oral cavity as long as you're chewing it and you swallow it, and then it's done. It stays in the stomach somewhere between 4 to 6, maybe 4 to 8 hours. It depends. The more fatty and protein-laden the food is, then the longer it stays in the stomach, which is why they say it's better to eat more protein than carbohydrates because it's more filling. But anyway, to get through the small intestine could take 6 to 8 hours, maybe even 12 hours. and you don't want to overwhelm any section. Overwhelming a section of the small intestine, like if you eat too much, causes things like diarrhea. So you don't want that. That is for sure. Let's see. Plus, you want the food stuff, the chyme, to move through the small intestine rather slower than it would through others because most absorption occurs in the stomach. So you want to make sure you absorb as much as possible or as much nutrients as possible from the food that you've eaten. There's some surface area modifications so we can maximize the surface area of the small intestine. First, there are villi, which are the finger-like projections that stick up. Plus, there's the plica circularis, which are the circular folds. They have crypts, which are parts of the cells of the mucosa that go down towards the submucosa. And these crypt cells produce the enzymes and hormones from the intestine. And then on the villi, there's also microvilli, and it's also called a brush border. The microvilli will touch the foodstuff it moves through and break things down as well using chemical digestion. They call that contact digestion. And so then there has to be a way to move the absorbed nutrients into the cardiovascular system. Most of it goes into the circulatory system, specifically the hepatic portal system, which we've talked about in the cardiovascular system. If anything that is a portal system means there are two series of capillaries. So the capillaries at the intestines move the nutrients to the capillaries of the liver. So it's all part of the hepatic portal system. And then we have lacteals, which are lymphatic capillaries to move the lipids that have been absorbed. So this picture is showing you the villus. Right here is the villus. The outer core of the villus, this is the simple columnar epithelium. They're also known as the absorptive enterocytes that go all the way around. Inside the core of the villus, we're going to find a nerve, part of a nerve. We're also going to find capillaries for the absorbed amino acids and carbohydrates. And then there's a lacteal, which is going to be the lymphatic capillary for the absorbed fats. In between each villus, we have an intestinal crypt. The cells of the intestinal crypt produce mucus, plus they also produce hormones like cholecystokinin. Sorry, I lost my train of thought there. And then if we take one of these cells, look at it, here's the enterocyte. They have microvilli on the apical surface, and the microvilli, also called the brush border, produces enzymes like enterokinase. All right, this is just showing the hepatic portal system. We have these capillaries in the digestive system taking the nutrients to the liver. And then we have capillaries in the liver. And then the blood that's leaving the liver is now nutrient poor, and it's also oxygen poor. So the blood that leaves through the hepatic vein joins with the inferior vena cava. Inferior vena cava then goes to the right atrium. And that's talking about the circulation of blood through the body. So blood coming down through the aorta, and then it goes through parts of the superior mesenteric artery, the gastric artery, so on and so forth. All of that blood's bringing some nutrients plus oxygen to the tissues of the gut. And then the blood that's leaving the gut is full of nutrients. So this is nutrient-rich blood. It's also oxygen-poor blood. This nutrient-rich blood goes into the liver processes the nutrients, stores the glucose as glycogen, takes the amino acids and turns them into proteins that are going to enter into the bloodstream. And then it takes the bilinogen that's coming from the spleen and changing it into bile. But then the blood that's leaving the liver has been cleaned, so to speak, of all the nutrients. And so now the blood that's leaving through the hepatic vein is oxygen poor and nutrient poor blood before it goes into the inferior vena cava. So these are all the types of secretions that are going to be emptied into the lumen of the intestine. We're going to have digestive enzymes. Most of the digestive enzymes that are going to be secreted into the duodenum, which is the first part of the small intestine, is coming from the pancreas. The most active enzymes for digestion come from the pancreas. The pancreatic amylase, pancreatic lipase, pancreatic nucleases, the zymogens, the inactive proteases. Anyway, so we have digestive enzymes being emptied into the small intestine. Bile is going to be emptied into the duodenum as well. Bile is coming from the common bile duct, which is coming from the hepatic and the cystic duct from the gallbladder. The cystic duct is from the gallbladder, and then the common hepatic duct is coming from the liver. They come together to form the common bile duct. The common bile duct goes all the way down to the duodenum and empties through the hepatopancreatic sphincter. I'm not telling you anything new. This is stuff that we went over in anatomy. You might need to review it just a little bit if you don't know what I'm talking about. Through the major papilla. And then we have bicarbonate, also coming from the pancreas. Bicarbonate is emptied into the duodenum because the gastric contents that are being emptied from the pyloric region of the stomach has a pH of about 1 to 2, but the pancreatic enzymes for digestion work best at a pH of about 8, so we have to raise the pH to 8. And that's what the bicarbonate is going to do from the pancreas. Then we have mucus coming from the intestinal crypts. Mucus is used as a lubricant, plus helps keep the chyme together as it's moving through the digestive tract. And then there's also going to be an isotonic saline secretion coming from the cells in the intestinal crypts, and that's so that the mucus doesn't stick to the lining of the alimentary canal. So this picture is showing you what's happening to the intestinal cells as they make the isotonic saline secretion. First is your gate at the basolateral surface of the cell. Remember, the basolateral surface is the surface that faces the interstitial fluid, which is your internal environment. The apical surface of the cell is going to be that part of the cell that faces the lumen, which is the external environment. So there is a gate that allows potassium, chloride, and sodium all to go into the cell. So potassium is going up its concentration gradient, chloride is going down its concentration gradient, and then sodium is going down its concentration gradient into the cell. Now the potassium will then go back out through a leak channel. The sodium will then be ejected back out into the interstitial fluid using the sodium-potassium pump. So chloride will pass through the cell, and then it's going to be emptied into the lumen of the alimentary canal. It's going to go down its concentration gradient into the lumen. Now wherever chloride goes, sodium and water is going to follow through the paracellular pathway between the cells. So now we have water, we have chloride, and we have sodium. So this is the saline solution that's going to line the lumen so that the mucus does not stick to the inner lining. Let's go over the function of some of the secretions and enzymes that are being emptied into the small intestine. Well, the endocrine secretions coming from the pancreas. We have insulin and we have glucagon. Insulin is released by the beta cells of the islets of Langerhans of the pancreas. Insulin is going to lower your blood sugar levels. So insulin is released whenever you have just eaten. So whenever your blood sugar goes up, insulin is released in order to put the glucose either into the liver or into your cells. Glucagon is a hormone that's released by the alpha cells of the pancreatic islets, and glucagon is released in between meals because it's going to raise your blood sugar. Glucagon will target the liver to break down the glycogen, and then it can release the glucose into the bloodstream. So the exocrine function of the pancreas is to produce the digestive enzymes. The cells that produce the digestive enzymes in the pancreas are called acinar cells, or acini, either way. They produce the pancreatic amylase, pancreatic lipase, and then the inactive protein enzymes called zymogens. The exocrine cells of the pancreas also produce sodium bicarbonate. These are going to be the ductal cells. These are the cells that line the duct. Remember, exocrine means this gland produces a secretion into a duct. Endocrine is a gland that produces a secretion directly into the bloodstream. So sodium bicarbonate is released by the duct cells, and then it is ejected into the duodenum. Now on the brush border of the absorptive enterocytes of the lining of the intestines, they have an enzyme called enteropeptidase. One of the inactive protein enzymes that comes from the pancreas is called trypsinogen. So enteropeptidase is an enzyme that converts trypsinogen into trypsin. And trypsin is going to activate the rest of the pancreatic enzymes. So it activates the rest of the pancreatic zymogens, the the phospholipase, the procarboxypeptidase, the chymotrypsinogen. We're not going to worry about the specific names. Just know that trypsinogen is an inactive protein enzyme that activates all of the other inactive protein enzymes, and it is because it's activated by enteropeptidase, which is an enzyme that comes from the intestinal brush border. The only digestive secretion that comes from the liver is bile, and bile it comes from bilinogen, which came from breaking down red blood cells in the spleen. So part of bile, don't have to worry about this too much, but there are bile salts, bile pigments, and some cholesterol. They're going to emulsify fats. So let's write that down, emulsify. That means to break down the fat. You can't use a watery enzyme to break down a lipid because lipids are made of fat. So you need a fat-based enzyme, which is going to be bile. Bile is based off of cholesterol, so it is fat-based. Now, bile is stored and concentrated in the gallbladder. The gallbladder sits right underneath the liver. There is a duct called the cystic duct that leads from the liver to the gallbladder. The gallbladder is going to take the bile, it's going to concentrate it by absorbing some of the water from it. And then whenever the food stuff, the chyme, hits the small intestine, it triggers the release of cholecystokinin. And cholecystokinin will then travel through the bloodstream because it is a hormone. It targets the gallbladder and it makes the gallbladder contract. When the gallbladder contracts, it will send the bile down through the common bile duct and eject just a little bit through the hepatopancreatic sphincter and into the duodenum. The functional unit of the liver is called the hepatic lobule. The hepatic lobule is made of plates of hepatocytes, and hepatocytes are liver cells. The liver cells are where we store glycogen. The liver cells are what produce the proteins, the plasma proteins. Now, the hepatic lobule is a six-sided figure, like a hexagon. At each corner of the hexagon is a triad of vessels. This triad of vessels include a bile duct. So the bile that's produced by the hepatocytes will exit through the bile duct. Then we have some blood going into the hepatic lobule. This is a branch off of the hepatic portal vein. And then we have blood coming in from the hepatic artery. So we have blood coming in through the hepatic portal vein, blood coming in through the hepatic artery. So as the blood goes through these plates of hepatocytes, it's delivering the nutrients, it is dropping off and delivering oxygen, picking up carbon dioxide, and then picking up the wastes. And then it's going to exit through this vessel in the very center, which is called the central vein. All of the central veins of each hepatic lobule will join together to form the hepatic vein. The hepatic vein leaves the liver and empties into the inferior vena cava. So bile is produced by the hepatocytes, exits through the bile duct, blood enters through the hepatic portal vein, hepatic artery, and then it exits through the central vein, which leads to the hepatic vein. This is a pretty good picture, and this is what I tried to explain in anatomy. So we have the liver, and these are the functions of the liver: glucose and fat metabolism, protein synthesis, hormone synthesis, produces urea, it detoxes the blood, and then we store nutrients, we store iron, we store glucose in the liver. Now let's look with hepatic portal vein. It's showing the direction of the blood, which is into the liver. What's carried by the hepatic portal vein is going to be blood filled with bilirubin. Bilirubin is going to be changed to bile. We have nutrients coming in from the hepatic portal vein from the gut. And then drugs that are circulating, these are going to be medications that we have taken orally, and then any type of foreign substances from stuff that we've taken orally as well. It's all being processed by the liver because the liver is going to detox your blood. Now let's look at the hepatic artery. Blood coming in through the hepatic artery, again going towards the liver. It is carrying blood that has bilirubin in it, has blood that has metabolites from hormones and drugs, and then also has nutrients in it. And then we have blood exiting through the hepatic vein. What's found in the blood of the hepatic vein, we have glucose, because the storage form of glucose is glycogen. Glycogen that is broken down by the liver is released as glucose. We have the plasma proteins, albumin, which creates the osmotic pressure. We have clotting factors like fibrinogen, and then angiotensinogen. Angiotensinogen we talked about in the urinary system. It breaks down to angiotensin 1. And then angiotensin 1 is broken down or changed into angiotensin 2. Do you remember which hormone changes angiotensinogen into angiotensin 1? In case you don't remember, it is called renin. So renin comes from the kidney, and it converts angiotensinogen into angiotensin 1. All right, also in the hepatic vein, we have urea and vitamin D. Vitamin D is a hormone that is produced, starts in the skin. When sun hits the skin, it makes a metabolite, which is then changed by the liver into vitamin D. And then stuff that we want to excrete comes out through the hepatic vein. It'll make its way to the kidney, and then the kidney is going to filter that out. And then let's look at the bile duct. The bile that exits the bile duct's got bile salts, bilirubin, some water ions, and some phospholipids. I skipped a few slides because we're not really going to get into the minutia of how fats are absorbed into the body. But most digestion occurs in the small intestine. So we have some digestion that occurs in the oral cavity, some digestion that occurs in the stomach, but most digestion occurs in the small intestine. Specifically, most digestion occurs in the duodenum, which is the very first part of the small intestine, which makes sense because that's where the protein enzymes are emptied into the small intestine through the duodenum. Most absorption occurs in the jejunum of the small intestine. So most digestion occurs in the duodenum, but most absorption occurs in the jejunum. Let's look at how carbohydrates are absorbed just a little bit, because we're all carb fanatics. But anyway, carbs can only be absorbed if they're monosaccharides. Monosaccharides are the smallest of the glucose molecules. And the monosaccharides are going to be glucose, fructose, and galactose. So unless it is glucose, fructose, or galactose, it's not going to make it into the intestinal lining and into the capillaries. So we have some common disaccharides. Maltose is basically two glucose molecules bind together. So maltose has to be broken down by maltase in order to produce the glucose molecules that can be absorbed. Sucrose is table sugar. Sucrose is broken down by sucrase and is broken down to a glucose and a fructose molecule. So sucrose, table sugar, cannot be absorbed into the lining until it's broken down into the glucose and fructose. Fructose is more like a fruit sugar. Then we have lactose. Lactose is a dairy sugar, so it comes from dairy products. The enzyme to break down lactose is lactase. So lactase breaks down lactose into glucose and galactose. If you are lacking the enzyme lactase, that means you're lactose intolerant, and you're unable to digest dairy products into the smallest forms in order to be absorbed. So that means the dairy products stay in the intestines. So if the dairy products stay in the intestines, what happens when it gets to the large intestine? Well, it creates osmotic pressure. So now the osmotic pressure inside of the large intestine is greater than the tissues, so the water stays in the large intestine and leads to watery stools or diarrhea. Or the other could happen. The body realizes there's dairy products in the stomach and it's just going to go ahead and vomit it out. All right, so what happens if we can absorb the smallest molecules as possible? There are gates located in the apical surface of the cell. We have the sodium glucose gate. We went over this in chapter 5. Sodium binds at the apical surface of the sodium glucose gate, which changes the affinity for glucose. So then both sodium and glucose bind at the gate, and they both enter into the cell. Sodium's going down its concentration gradient. Glucose is actually going up its concentration gradient. So then the sodium will be exchanged for potassium in a sodium-potassium pump. But then the glucose will then go down its concentration gradient through a glucose channel at the basolateral surface, and then the glucose will go into the capillaries. And that's how we absorb that. But then there's also some glucose channels that are in the apical surface that is specific for fructose. So fructose and can also enter through a fructose gate, and it goes up its it's going to go down its concentration gradient and then into the system. All right, this slide is also easy to read. We're talking about how we absorb certain vitamins and minerals and other ions. The fat-soluble vitamins have to be absorbed with fats. The water-soluble vitamins can be absorbed by mediated transport. Don't worry about these specific parts. But we talked about vitamin B12 earlier. Vitamin B12 can only be absorbed if it's bound to intrinsic factor. An intrinsic factor comes from the stomach. It comes from the parietal cells. So unless vitamin B12 is bound to intrinsic factor, you're not going to absorb it through the intestines. And then we have iron and calcium. Iron and calcium have to be absorbed through specific iron and calcium channels. So that absorption can be actively regulated by either creating or destroying the iron or calcium gates. Now this is when we talked about the parathyroid hormone, and then we have calcitonin, which is a thyroid hormone. Calcitonin will decrease your blood calcium levels, and one of the ways it'll do that is by blocking the calcium channels in the intestine so that you don't absorb that much calcium from the food stuff that you ate. Then we have parathyroid hormone, which does the opposite. Parathyroid hormone will activate calcium gates in the apical surface of the lining of the alimentary canal so that you can absorb more calcium into your system. The last phase of the intestinal phase occurs in the large intestine. So if we just recall the parts of the large intestine. First, the valve between the large intestine and the small intestine is called the ileocecal valve. So ileocecal valve, that is the valve between the small and the large intestine. It controls how much chyme passes into the large intestine. The main function of the large intestine is to absorb water and electrolytes. So anything that passes into the large intestine is no longer called chyme, it is now feces, because basically this is going to be indigestible residues that are going to be eliminated. But the body wants to reclaim as much water as possible. So by the time it enters into the large intestine, there's only about 1.5 liters of chyme left, and then most of that is absorbed in the colon, and so that only about 0.1 liters of water is going to be lost with the feces. Anyway, so the very first part of the small intestine is called the cecum, and then the ascending colon, and then there's a flexure called the hepatic flexure, and then we have the transverse colon, and then there's another bend called the splenic flexure, and then there's the descending colon, which leads to the sigmoid colon, which leads to the rectum, and the rectum leads to the anal canal. There's a piece of lymphatic tissue that's attached to the cecum that's called the appendix. So the appendix is lymphatic tissue which has a function like the rest of the lymphatic system, but the appendix, if it becomes really heavily involved and infected, then it'll have to be removed before it bursts and it just floods your abdominal cavity with fecal material, which can be poisonous to those other tissues. Now, the arrangement of the longitudinal muscle in the large intestine is called tinea coli. So tinea coli is a longitudinal muscle, and it creates pouches, which is called haustra. When it comes to movement or motility in the large intestine, there's not that much peristalsis. Peristalsis pretty much stops. After that, we still have some segmental contractions for mixing the food, the stuff that's left in the large intestine. And now this calls it chyme, but I'm telling you right now it's not called chyme, it's called feces. The type of movement instead of peristalsis is called mass movement. And mass movement is going to move the feces through most of the large intestine. We only have 2 to 4 mass movements per day, depending on your meal plan or how often you eat meals. But they only happen 2 to 4 times a day, and that's it. Peristalsis, when it comes to everywhere else in the alimentary canal, is pretty much 24 hours, happens all the time. So mass movement only 2 to 4 times per day. There are some reflexes associated with the large intestine. We have the gastrocolic reflex. The gastrocolic reflex means that as food enters into the stomach, which will be the gastric part of the gastrocolic, then it opens the ileocecal valve, and it creates a mass movement. That is the gastrocolic reflex. The defecation reflex is whenever pressure is applied from the feces onto the rectum. Once there's pressure from the feces on the rectum, then a signal is sent to the small intestine, which then sends a signal to the rectum to open the internal anal sphincter. Now you have 2 anal sphincters, internal and external. The internal anal sphincter is involuntary. The external anal sphincter is voluntary, and that way you can hold it until you're able to get to the bathroom. So the defecation reflex starts whenever the feces stretches the rectal wall. The rectal wall then sends a signal to the spinal cord, which then sends a signal to the brain to let you know that you have to go. Now there is a bacterial flora that lines the lumen of the large intestine, and this bacteria can help break down some undigestible materials. It also helps absorb certain vitamins, like vitamin K can be absorbed through the small intestine because of the bacteria, further breaking down some of the food stuff that's still left in the large intestine, plus some vitamins. And then the bacteria also creates a gas, which is known as flatus. Diarrhea is whenever we have loose stools that are watery stools, and if you have diarrhea enough, it can cause dehydration. So here's a picture showing you the large intestine. Right here's the ileocecal valve, the valve between the ileum, the last part of the small intestine. Here's the cecum, the first part of the large intestine. Right here's the appendix, which hangs off of the cecum. The foodstuff that enters here will then be passed up through the ascending colon, then around the hepatic flexure through the transverse colon, then around the splenic flexure into the descending colon, and then it goes to the sigmoid colon, and then out through the rectum, and then the anal canal. Now you'll see that there's a ribbon of muscle in the very center. This is the longitudinal muscle. So it's not around the entire circumference, it's just located in these areas right here. This is the tinea coli. These muscles, whenever they contract, they shorten part of the large intestine and they push the feces up. So during a mass movement, the feces that started right here will be pushed up and over, all in one type of movement. And that starts with the gastrocolic reflex. And then we have some anal canal or anal sphincters. There's the internal anal sphincter, which is involuntary, and we have the external anal sphincter, which is voluntary, so that you're able to hold it in the opening to the outside, which is the anus. Remember that the rectum is lined with stratified squamous epithelium. Most of the rest of the alimentary canal is lined with simple columnar epithelium. There are no villi, and the cells of the lining are called colonocytes or colonocytes. Now I want you to point this out right here because I think it's interesting. There are 3 valves in the rectum called rectal valves. These 3 rectal valves will help hold on to the feces so that you can pass gas without pooping in your pants. All right, I'm going to leave it on that note. I don't have anything else to say about this chapter. I'm not going to worry about this last slide. So if you have any questions about this chapter, please email me. Thanks.